Cloning. What exactly is cloning? Why exactly is cloning? What's good about it? What's bad about it? Are we doing it now? Yeah, actually, we are, but there's some things I want to cover first. Clones are organisms that are exact genetic copies. Every single bit of their DNA is identical. But clones can also happen naturally. Identical twins are just one of many examples, or they can be made in the lab. Some examples of natural cloning in the wild would be all bacteria. The majority of fungi and many species of protists reproduce asexually to produce genetic clones. While most plants reproduce sexually, they also possess methods of asexual reproduction, like vegetative propagation. Certain animal species can also reproduce asexually via a variety of different mechanisms. Many people first heard of cloning when Dolly the sheep showed up on the scene in 1997. Artificial cloning technologies have been around for much longer than Dolly, though. There are two ways to make an exact genetic copy of an organism in a lab: artificial embryo twinning and somatic cell nuclear transfer. Artificial embryo twinning is a relatively low-tech way to make clones. As the name suggests, this technique mimics the natural process that creates identical twins. In nature, twins form very early in development, when the embryo splits in two. Twinning happens in the first days after egg and sperm join. While the embryo is made of just a small number of unspecialized cells, each half of the embryo continues dividing on its own, ultimately developing into separate, complete individuals. Since they developed from the same fertilized egg, the resulting individuals are genetically identical. Artificial embryo twinning uses the same approach, but is carried out in a petri dish instead of inside of the mother. A very early embryo is separated into individual cells, which are allowed to divide and develop for a short time in the petri dish. The embryos are then placed into a surrogate mother where they finish developing. Again, since all the embryos came from the same fertilized egg, they are genetically identical. Somatic cell nuclear transfer is the next one. Now, this one's a little more interesting. Somatic cell nuclear transfer, or SCNT, also called nuclear transfer, uses a different approach than artificial embryo twinning, but it produces the same result: an exact genetic copy or clone of an individual. This was the method used to create Dolly the sheep. Now, what does SCNT mean? Let's take it apart a little bit. A somatic cell is any cell in the body other than sperm and egg. The two types of reproductive cells. Reproductive cells are also called germ cells. In mammals, every somatic cell has two complete sets of chromosomes, whereas the germ cells have only one complete set. Next, nuclear. The nucleus is a compartment that holds the cell's DNA. The DNA is divided into packages called chromosomes, and it contains all the information needed to form an organism. It's a small difference in our DNA that makes each of us unique. And finally, transfer. Moving an object from one place to another. To make Dolly, researchers isolated a somatic cell from an adult female sheep. Next, they remove the nucleus and all of its DNA from an egg cell. Then they transfer the nucleus from the somatic cell to the egg cell. After a couple of chemical tweaks, the egg cell with its new nucleus was behaving just like a freshly fertilized egg. It developed into an embryo, which was implanted to a surrogate mother and carried to term. The transfer step is most often done using electrical current to fuse the membranes of the egg and the somatic cell. The lamb, Dolly, was an exact genetic replica of the adult female sheep that donated the somatic cell. She is the first ever mammal to be cloned from an adult somatic cell. Now, how does SCNT differ from the natural way of making an embryo? Natural fertilization, where egg and sperm join, and SCNT both make the same thing: a dividing ball of cells, called an embryo. So what exactly is the difference between the two? An embryo's cells all have two complete sets of chromosomes. The difference between fertilization and SCNT lies in where those two sets come from. In fertilization, the sperm and egg have one set of chromosomes each. When the sperm and egg join, they grow into an embryo with two sets, one from the father's sperm and one from the mother's egg. In SCNT, the egg cell's single set of chromosomes is removed. It is replaced from the nucleus from a somatic cell, which already contained two complete sets of chromosomes. So in the resulting embryo, both sets of chromosomes come from the somatic cell. You may have heard about researchers cloning or identifying genes that are responsible for various medical conditions or traits. What's the difference? When scientists clone an organism, they are making an exact genetic copy of the whole organism as described above. When scientists clone a gene, they isolate and make exact copies of just one of an organism's genes. Cloning a gene usually involves copying the DNA sequence of that gene into a smaller, more easily manipulated piece of DNA, such as a plasmid. This process makes it easier to study the function of the individual gene in the laboratory. Now we know what cloning is and how we do it, we can talk about some of the advantages of cloning. Why should we do it? Well, cloning allows farmers and ranchers to accelerate the reproduction of their most productive livestock in order to better produce safe and healthy food. Cloning reproduces the healthiest animals, thus minimizing the use of antibiotics, growth hormones, and other chemicals. Consumers can benefit from cloning because meat and milk will be more healthful, consistent, and safe. Most of the foods from cloning will be from the offspring of clones that are not clones themselves, but sexually reproduced animals. Cloning can be used to protect endangered species. For example, in China, panda cells are being kept on reserve should this species number be threatened by extinction. They also allow research into genetic disorders by having a control study or replicating an animal scenario to try and drive answers from that. However, with all the good it may cause, there's some ethical concerns we need to take into account too. Most of these are theoretical in nature, as human therapeutic and reproductive cloning are not commercially used, but focused rather on animals that are now being cloned in laboratories or in livestock production. Advocates support the development of therapeutic cloning in order to generate tissues and whole organs to treat patients who otherwise cannot obtain transplants, to avoid the need for immunosuppressant drugs and to stave off the effects of aging. Advocates for reproductive cloning believe that parents who cannot otherwise procreate should have access to the technology. People against cloning have concerns that technology is not yet safe and that could be prone to abuse, either in the form of clones raised as slaves or leading to the generation of humans from whom organs and tissues would be harvested. They've also raised concerns about how cloned individuals can integrate with families and with society at large. Religious groups are divided, with some opposing the technology as usurping God's place, and to the extent those are used, destroying a human life. Others support therapeutic cloning's potential life-saving benefits. Cloning of animals is generally opposed by animal groups to the number of cloned animals that suffer from malformations before they die, and while food from cloned animals has been approved by the FDA, some animal health groups do not like that. With that, what do you think about cloning? Is it safe? Should we keep using it? Should we clone humans? I'll let you decide.